Learning target 4.7 is about the structure of DNA, and you guys have been doing this activity in class to explore what the structure of DNA looks like. And so all the specifics of that structure, we're going to come back and talk about together later. So learning targets 4.7a through F, we will come back and talk about later, but we're going to skip over those for now. And we're going to go straight to learning target 4.7g. And learning target 4.7g is all about how did we first discover that DNA is the important genetic material within your cells. So we all know that DNA is that important information that has to get passed from cell to cell in order for that new cell to know how to function. It has to get passed from parents to their children to pass all of those traits onto their children and make their kids have the particular features that they have. But how did we first figure out that DNA was the molecule in our cells that does that? We're going to go through experiments from 3 different scientists and famous experiments that were done that were kind of the steps in helping us to figure out just how important DNA is, and that it is that material inside of your cells that has to be faithfully passed on to new cells in order for those new cells to have the properties that they have and be able to function. So the first experiments we're going to look at were done by a man named Frederick Griffiths. So these are Griffiths experiments. And Griffiths is going to be working with some different types of bacteria, and he injects those bacteria into mice and basically sees what happens to the mice and makes some conclusions from that. So first off here we have the strain R bacteria, and if you inject R strain bacteria into a mouse, the mouse is actually totally fine. It's not a type of bacteria that's bad or deadly in any way. It's what we call non-virulent, and so that mouse is totally fine. Now, on the other hand, he has a different strain of bacteria, what we call here strain S. It's a different type of bacteria. And if you inject that one into a mouse, so here's strain S, inject that into the mouse, and the mouse dies. So 2 different types of bacteria. One's deadly, one's not. Now, for his next experiment, he takes that S bacteria that we know is supposed to be deadly to the mouse, and he now does what we call heat kills it. Basically, he takes that bacteria and heats it up to a really high temperature. And as you guys should already know from stuff we've talked about earlier this year, if you take living things and heat them up to high temperature, that's a problem because it's going to cause their proteins to denature. And it basically then kills those bacteria. So if you take those bacteria that are normally deadly, but you kill the bacteria, well, you can inject those dead bacteria into the mouse. They're not going to have any effect. So in this situation, the mouse is totally healthy. In his last experiment, he actually mixes 2 of these different bacteria together. So he takes that R strain bacteria, and these ones are alive, but we know that a living R strain doesn't do anything to the mouse. And then he takes some of this heat-killed S strain that also didn't kill a mouse, and he mixes those 2 things together. So he's taking these 2 things that independently left the mouse healthy, injects both of them together in a mouse to see what happens. And what happens is the mouse actually dies. So even though these 2 things independently weren't able to kill the mouse, when they're mixed together, all of a sudden they affect the mouse. And even more surprisingly, if you take that dead mouse and you take a sample of his blood and you look in his blood, you can actually find some living S strain bacteria in his blood. And so what Griffiths figured out is that even though this S strain bacteria is dead, parts of that S strain bacteria are still going to be there. They may not be functioning and working and living things, but you're still going to have proteins there, and you're going to have DNA there, and you're going to have some carbohydrates that are left behind from those cells. So he figured something from these dead cells must actually get passed to these living cells and turn these living R cells into S cells. So Griffiths figures out that there's some material that can be transported from one cell to another cell, or even just from the remaining parts of dead cells to another cell, that can actually change how those cells function and what their properties are and give them brand new properties. In this case, that property making it those cells can kill that mouse. Now, Griffiths doesn't figure out that DNA is that important information that has to be transferred. We all know today that DNA is that important information in your cells that tells them how to function and gives them their properties. Griffiths didn't figure that out. But he starts to figure out that there has to be some important controlling information, and that information could be passed from cell to cell. This same type of experiment that Griffiths did, we're going to basically repeat the same experiment, or someone by the name of Avery is going to repeat the same experiment, but he's going to make one little addition that's going to answer some more questions about our genetic material. So the next experiment we're going to look at is an experiment done by Avery. This is Avery's experiment. And Avery's going to repeat what Griffiths did. He's going to take that R strain that was a non-killer, the heat-killed S strain, which now that it's dead, doesn't kill the mouse, and he's going to inject both of those into the mouse. He's going to do that. We're going to show 2 different times here. He actually did some additional experiments as well, but we're going to look at these 2. But he made one addition to what Griffiths did. When he injected those things into the mouse, he added something else. In one situation here, he adds what's called a nuclease. And in the other situation over here, he adds what's called a protease. And a protease is something that can destroy proteins. And a nuclease is something that can destroy nucleic acids, like DNA. So he takes those things, injects them into the mouse, and here's what he finds. He finds that when you add a nuclease, the mouse is healthy. And when you add the protease, the mouse dies. And this is what helps us to realize that DNA is that important information that gets passed from cell to cell. Because what Avery figured out is that if DNA is what's being passed from those heat-killed S strains into R strain cell, if that's the important genetic information that has to be passed, then what he figures is happening is that DNA is supposed to be passed from this cell to this cell. But if I add a nuclease, that DNA gets destroyed. And if DNA gets destroyed, then there's nothing to be passed into these cells. And so now I just have my normal R strain, which were never a killer, and so they're not a killer type of cell and the mouse lives. If I had a protease, on the other hand, who cares if I destroy the proteins? What's important is that the DNA from these cells gets passed to these cells. And as long as that DNA can be passed, which that DNA is totally fine here, because I only used a protease, as long as that DNA can get passed, then the DNA from those S cells gets transferred into those R cells and gives those R cells new properties. And that property that it's gotten from the S cells is that it can now kill the mouse. So Avery does this great experiment that helps us start to realize that DNA is that important information that can and needs to be passed from cells to cells and gives those cells the particular properties that they have. Now, Avery's experiment is pretty cool and straightforward, but when it was done, a lot of people still weren't really convinced. Because even though we haven't talked a ton about the structure of them yet in class, what people knew at the time is that DNA is a relatively simple molecule and proteins are a lot more complicated. And people found it hard to believe that it's the simple molecule, the DNA, that can do the super, super important stuff. There were some people that figured DNA is just too basic and simple a molecule to be this important and control all these things happening in cells. And even though the evidence wasn't supporting it, there were some people that held on to the idea and wanted to keep believing that it was actually proteins that were the important genetic information. So in addition to Avery's experiment, another experiment gets done a number of years later that reinforces Avery's findings and finally gets more people to start believing that DNA is in fact that genetic material. And those experiments were done by Hershey and Chase. So this is the Hershey and Chase experiment. And what Hershey and Chase did was they used these right here, which are viruses. Okay? And viruses are really cool for testing this question because what people are trying to figure out is it the DNA or is it the protein? Which of those is the important genetic information? Well, really simple viruses like this, some viruses are more complicated, but really simple viruses like this are only made up of 2 things. They're made up of DNA and they're made up of protein. So if those are the things we want to test to figure out what's the genetic information, viruses seem like a great tool we can use to test that. And what viruses like to do is they infect cells. So these viruses here are going to infect a bacterial cell, and the viruses essentially take over the bacterial cell. They start controlling that bacteria and making that bacteria do things for the virus. So again, we're going to do this experiment twice, and we're going to do slightly different things each time. So in one experiment, they took the protein, and so all this outer stuff here that's highlighted in pink, that's the protein. And the DNA is right there inside of this protein up here in what we call the head region of it. So they took just the protein and they made it radioactive. They basically labeled the protein. So the protein's labeled, but the DNA is not. And then they do a corresponding experiment where they label the DNA. So the DNA here you can see is glowing in blue, but they didn't label the protein. So in one, the protein's radioactive, and the other DNA is radioactive. You take those viruses and you let them infect the bacteria, and then you come back a while later and you look at those bacteria. And their reasoning was, if viruses are basically able to take over and start controlling the bacteria, then whatever that genetic information is, which controls cells and helps us figure out how to function, must end up inside of those bacteria. So they're trying to check to see what gets inside of those bacteria. And what they found is that in this situation where they had labeled the protein and made the protein radioactive, they don't see any radioactivity inside of their cells, meaning that protein never makes it into the bacterial cell. On the other hand, when they made the DNA radioactive, they do see some radioactive signal coming from the bacteria, meaning that DNA did get inside of the cell. And so their big conclusion then is DNA must be the genetic material. If DNA can get inside of a bacterial cell, but protein can't, and we know that those viruses are somehow taking over those bacteria, then it has to be the DNA that's inside of that cell that's basically starting to control and take over that bacteria. Therefore, DNA must be the genetic material. Okay? That's learning